So we just talked about a lot of situations where things can go wrong. But under normal circumstances, signal transmission through the neuromuscular junction results in the muscle action potential traveling down the sarcolemma. Muscle fibers can then use that action potential to initiate the process of contraction. This transduction of an electrical signal into a mechanical force is referred to as excitation-contraction coupling, and that's what we'll be talking about in this video. Now, in order to understand the process, we have to think back to the internal structure of the muscle fiber. And there are really three things to point out from these diagrams that we're looking at here. The first is that the muscle fiber is very, very densely packed with myofibrils, and that leaves very little cytoplasmic space. The second thing is that the plasma membrane, sarcolemma, represented in yellow in both of these diagrams, has a very specialized structure in muscle. So in addition to being at the surface of the cell like we would expect, it also travels deep down into the inner part of the muscle fiber, creating a structure called a T-tubule that sort of resembles a canyon. You can see this especially clearly in the image on the right. That dark yellow pit is the T-tubule. The T-tubules allow rapid exchange between the interstitial fluid and the deepest parts of the muscle fiber. So rapid exchange between inside and outside, even deep within the cell. And this is necessary because muscle doesn't have a lot of cytoplasm for molecules to diffuse through. The third thing that we see is the sarcoplasmic reticulum, which is represented in these diagrams in blue, is extremely well developed inside of muscle. So the sarcoplasmic reticulum is the main calcium storage organelle inside of muscle. And remember that calcium is at a relatively high concentration in the interstitial fluid, an extremely low concentration in the cytoplasm, but it's highly concentrated inside of these intracellular stores inside of the sarcoplasmic reticulum, or SR. So the calcium that muscle needs for contraction is actually getting released from these intracellular stores in the SR. Now, before we move on from this slide, I also want to point out that there's a close relationship between the T-tubule and the sarcoplasmic reticulum. You can see that on either side of the T-tubule, you have the membrane, or the feet, of the sarcoplasmic reticulum. This is known as the triad. Now, even though the T-tubule is the plasma membrane and the sarcoplasmic reticulum is an organelle inside the cell, the two of them are located so close together that they're practically touching each other. There's very, very little cytoplasmic space between them. So this diagram here ties together all of the events at the neuromuscular junction, which we talked about before, with the triad that I've just mentioned. And it's this diagram here that we're going to use to walk through the steps of excitation-contraction coupling. So excitation-contraction coupling begins with excitation. That's the change in membrane potential that's created at the neuromuscular junction that we've just talked about. When the action potential leaves the motor end plate and travels from membrane segment to membrane segment to membrane segment down the sarcolemma, eventually it is going to enter the T-tubule. And there's a voltage-sensitive protein in the T-tubule called DHP, which stands for the dihydropyridine receptor. We'll always abbreviate it DHP. You can see that really close relationship between the T-tubule and the sarcoplasmic reticulum here. Now, on the sarcoplasmic reticulum membrane, there's a calcium channel called the ryanodine receptor, which is abbreviated RYR. Now, even though DHP and RYR are on two different membranes, they're so close together that people often will describe them as being physically connected to each other. So when the action potential travels into the T-tubule and depolarizes the membrane around the DHP will change conformation. And this pulls open the ryanodine receptor and allows the highly concentrated calcium inside the sarcoplasmic reticulum to move down its concentration gradient into the cytosol. I often think of this process as being like pulling open the drain on a tub. Your hand is the DHP and the stopper is the RYR. The water would be the calcium. Now, in this step, we've used our action potential, which was carried by sodium, to give us all the calcium that's necessary for muscle contraction. That calcium can then bind to troponin and move the tropomyosin out of the way and initiate the process of filament sliding that we've already talked about in the previous videos. This gets us through the contraction part of excitation-contraction coupling. Now, contraction is always the goal, but relaxation is equally important. I don't know about you, but I like to use my muscles more than once. So you don't just want to contract your muscles one time. You want to relax so that you can contract them again and again and again. In order for that to happen, calcium has to return to its resting levels. We have to reset the concentration gradient. But keeping in mind the relative concentrations, calcium's higher outside the cell and in the sarcoplasmic reticulum and low inside the cytosol. If we try to move the calcium out of the cytosol, then we're moving it against its concentration gradient. So that means that we have to use active transport. One helpful thing to remember here is that calcium always goes back to where it came from. The calcium for muscle contraction came out of the sarcoplasmic reticulum through the ryanodine receptor, so it needs to return to the sarcoplasmic reticulum. But it can't do that through a channel. Instead, it does it through an active transporter known as SERCA, or the sarcoendoplasmic reticulum calcium ATPase pump. That's a mouthful to say, so that's why most people usually just call it SERCA instead. But if you do remember the full name, that really tells you exactly what it is. It's an active transporter. It's located on the membrane of the sarcoplasmic reticulum, and it uses ATP to move calcium against its gradient back inside the SR. This is excitation-contraction coupling, start to finish.